Good morning, good morning, good morning. Yeah, good morning guys. YouTube पे नहीं चालू हुआ है क्या रवि? Started ना? अच्छा. Yeah guys, so finally the date is out guys, ठीक है? 21st June. Very well predicted by our chairman sir. हाँ. Good morning, good morning, good morning. Come on, let's start. We have to study two topics today. We have to talk about the frog chapter, and then we have to also study the breathing and exchange of gases. Good morning, good morning, good morning, students. So let us start. First, we will start with the frog topic. Okay? So let's quickly start with the frog topic. अबे कहाँ गया frog? Sir, voice is not coming. Voice is not coming. Hello, everyone. Hello. No, cockroach not required. Cockroach not required. Cockroach not required. Cockroach not required. Cockroach not required, guys. Okay. Voice is coming. Voice audible, everyone. Voice audible, everyone. Okay. So, sir, video is turned off. No, video is very well off. Sorry, it's on only. Sir, when will the classes conducted for incoming seniors? For incoming seniors, it will be notified, guys. Sir, it will be notified. Yeah, I will be covering both frog and breathing today. So come on. Let us continue. So today's topic is tissue, frog, and breathing and exchange of gases. I will request you all to revise tissue, epithelial and connective tissue from the NCERT lines. We will directly jump on and start with the frog topic. Okay. So in the first paragraph, in the frog topic, in the first paragraph, they are talking about, in the preceding chapters, you studied about the unicellular and multicellular organisms. For unicellular, a single-celled organism, everything is done by the cell itself, respiration, digestion, all the metabolic process. But as the evolution happened, we got the complex multicellular animals in which the functions got distributed, or I will say there was division of labor, which you studied in animal kingdom also. There was first cellular organization, then tissue, then organ, and then organ after followed organ system organization, where we are seeing the division of labor. And then they are talking about little bit about the tissue definition here. They are talking about the tissue definition. So from this paragraph, you have to remember the tissue definition. Then directly moving to this paragraph, the second paragraph is talking that complex animals, they consist of four basic type of tissues that we already know. Epithelial, connective, nervous, and muscular tissue is there. Like any organ, if I talk about stomach, or if I talk about heart, is it okay to say that heart is only made up of cardiac muscles? Or stomach is only made up of epithelial cells? Is it okay to say like that? No. Any organ, if you look, all the four tissues are present. Like for example, if you talk about stomach or you talk about lung, heart, all these organs contain all the four different types of tissues, and they are performing a common function together. Okay? Then organ and organ system. The basic tissues you have learnt in the earlier classes. Okay, how they got organized, the organization you studied in the animal kingdom. So I will be skipping this part, going directly here. See, here they have written an example. For example, our heart consists of all the four types of tissues. Like heart doesn't only contains the cardiac muscles, right? Cardiac muscles, I can say it is the major tissue present, which is helping in the contraction, because the main function of heart is pumping of the blood. For that, you require the cardiac muscles. But the same heart, which is lined by pericardium, which is made up of epithelial tissue, now the pericardium, and then connected to the inside lining of the heart, that is connected by connective tissue. The heart is pumping the blood, that is connective tissue. Heart also is regulated by the sympathetic and the asympathetic nervous system. So there is neural tissue as well. So you can see here all the 4 different types of tissues are there. Are we clear? So we also notice after some careful study that the complexity in organ and organ system display certain discernible trend. Now, what is the meaning of this discernible word here? Discernible means visible. Discernible means visible. So as you study from the lower organisms to higher organisms, you can see a clear visible trend that complexity is increasing. But why is the complexity increasing? Complexity is increasing to divide the functions for better division of labor and better performance. Okay? Arey, don't be scared, yaar. Come on. Don't be scared. What you will get by getting scared? Nothing to be scared. Same level of the paper is going to come. Whatever paper you wrote, okay, you find it very easy or moderate, whatever it was. theek hai? Same type of paper is going to come. Everything is in the mind. If you constantly tell your mind that, okay, there will be a challenging, tough paper, you will get that. But if you train your mind, no, I know everything, I have revised well. Again, I am confident I am going to repeat my same performance. Okay? Then you will get that confidence. And please, you have written so many grand tests. Now imagine that NEET test which happened, that was also a kind of a grand test only. Yeah, that came a little easier or moderate level. Same kind of paper is going to come. Okay? So just chill and revise the NCERT, revise the important points, whatever the teachers are telling. Go through that, write your examination. Don't think anything else. There's no time to cry or whine about it. Just face the situation, study, revise, and write the paper. And again, get your admissions, okay? This was not the way I wanted to meet you all. I was thinking about the reunion, but theek hai. Okay, we met again. So let's continue. Now, next part, morphology. theek hai? Okay, now morphology. Scored 350 in the last exam, sir, in bio. No problem, Shikara. Again, you are able to do that. No problem. A warrior is not defined by just winning one war. So there is a second war for you to define yourself again. Okay, so chill. Come on. Now, no distractions. Okay, I don't want to give any motivation. We have to study a lot. So please focus. So morphology. What is morphology? When you study the external visible features, that you called as morphology. And when you study the internal part, that is called as the anatomy. Externally you are studying anything, that is the external morphology. External visible structures, that is morphology. Internal structure, when you are studying, you call it as the anatomy. And when you are studying the cells and the tissues, you are observing the tissue. What you call them as? Tell me. When you are studying the tissue, then what you will call it as? External morphology, internal anatomy. And when you study tissue, what it will be called as? It will be called as the it will be called as histology. It will be called as histology when you study the tissue. Come on, let's talk about frog. Frog lives on both land and freshwater. theek hai? Freshwater is very important. Remember, to trick you, they might give you some marine water or something. Be focused. Amphibia is a class belonging to phylum Chordata. Most common species of the frog we are discussing here is Rana tigrina. What is the order? Can anyone tell me the order of this frog here? Or frog order? That comes from the name. They are not having their tail. They are not having their tail. Yes, Karthike, right answer. Anura is the answer. Anura means not having the tail. So just remember the order Anura, okay, because they are not having the tail. That might be asked as well. Okay, now they are cold-blooded animal. They don't have a constant body temperature. If the weather outside is very hot, their body temperature will also become hot. If the weather is outside cold, their body temperature will also become very cold. So during extreme hot and extreme cold weathers, they go under hibernation and estivation. In winter, winter sleep, hibernation, summer sleep, estivation. They cannot regulate their body temperature. That's why they are cold-blooded animals. Another word for cold-blooded animals, we will say they are poikilotherms. They are what, guys? They are poikilotherms. Then frog can also change their color. In their grass, they will appear a little greenish. In the land or in the soil, they can little bit change the color, and they can go under camouflage. So what is the line written here? They have the ability to change the color and hide from their enemies, their predators. They are hiding from their predators. That is called as the camouflage. And this protective coloration is called mimicry. Remember this line as it is: hiding from the enemy, okay, adapting to the similar color to their surroundings, camouflage. And this protective coloration is called as mimicry, mimicking someone, okay. Moving ahead, you also notice you are not seeing the frogs in peak summer and winter because they are going under hibernation in winter and summer estivation. Okay, moving ahead, talking further about morphology. When you have touched the frog skin, it is very smooth and slippery. Why it is very smooth and slippery? Because there is a presence of mucus. So in the summary lines, if you go and check the summary lines, there it has written the frog skin has the mucus glands, which is constantly producing mucus and it is keeping it wet. Why it is keeping it wet? Because the majority of the time the frog is breathing via the skin. That is the cutaneous respiration. That's why we have to keep it moist. Okay, the dorsal side of the color is generally olive green with dark irregular spots. Okay, on the ventral side, on the belly side, it is pale yellow color, and frog never drinks water. It always absorbs through the skin. Okay, mostly through the ventral part of the skin, it is mostly absorbing the water. It will never drink. Okay, now body of the frog is divisible into two parts: head and trunk. Neck ledu, tail ledu. Neck, why not there? Some question is asked in the long term, or some questions were there previously when we started this chapter. Neck absence create balance. So imagine a frog with a neck and it is jumping. So what will happen? The neck may create some imbalance. So that's why neck is absent, okay? Helps in better balancing the body. Above the mouth, there is a pair of nostrils, as we can see in the diagram. Eyes are bulging out. Eyes are covered by a nictitating membrane. What is the function of nictitating membrane? The nictitating membranes are protecting the eyes in the muddy waters when they are swimming. On the either side of the eyes, we can see the hearing structure is there. The tympanum is there, which receives the sound vibrations or the waves. Then we talk about the forelimbs and the hind limbs. Forelimbs and hind limbs, okay, what they are used for? They are used for swimming, walking, leaping and burrowing. Hind limbs have 5 digits. Forelimbs have 4 digits. Do they have nails? Do they have nails? Do frog have nails? Tell me yes or no. And talking about the webbing, talking about the webbings, the feet have webbing. Feet have web digits and that help in swimming. Forelimbs, webbing ledu. Hind limbs are the feet which is having the webbing. Are we clear? Yes. Sir, since they can change temperature, they do not change temperature, Ram Krishna. Ram Krishna, they do not change their temperature. Their temperature is not regulated. So whatever is the outside temperature, that will be their body temperature. So if it becomes extremely hot and cold, they have to hide. Okay, next. Nails ledu. Nail not there. Okay, nail, claw, nothing is there in frog. That is nails and claws are the features of the reptiles, I can say. Claws basically. Okay. Then feet are having web digits that help in swimming. Now certain PYQs are there. They are writing the webbing is directly proportional to the amount of time spent in the water. Okay, that is also a PYQ old question, that the more amount of time the frog spends in the water, their webbing further develops and that may help in swimming. Now frog exhibits sexual dimorphism. Okay, very important. That also came in this NEET exam, that male frog and female frog, they have certain distinguishing points. So in a species, if there are differences in the male and female, that is called as sexual dimorphism. Like humans also, there is sexual dimorphism, the differences between male and female. One difference is the vocal sac. The vocal sac is present in the male, absent in the females. Okay, so it doesn't mean that female cannot produce sound. Male and female frog both have vocal cords. Vocal cord is something else which is not mentioned in NCERT, so listen carefully. Vocal cords are present in both male and female. So if in my both hands, if I have one male and one female, if I just press them, squeeze them little bit, both will produce sound. But tell me, who will produce a larger sound? Who will produce an amplified sound? A larger sound will be produced by the male frog that is having the vocal sac. So vocal sac is kind of helping in the amplification of the sound, okay? Croaking or the mating call during the rainy season when their mating is there. Are we clear? So vocal sac present in male frog and the copulatory pad, which develops during the breeding season, especially in the rainy season, on the first digit of the forelimb. Copulatory pad on the first digit of the forelimbs. Are we clear with this? Understood? Moving ahead, now talking about anatomy. Okay, this was all about the external morphology. Now we will talk about anatomy. So talking about anatomy, the body cavity of the frog accommodates mostly all the organ systems like us: digestive, circulatory, respiratory, nervous, excretory, everything is present. Now talking about digestive system is made of alimentary canal and digestive gland. What is the important point here? The important point is alimentary canal is short because frogs are carnivorous. Okay, so it means the animals who are herbivorous, their alimentary canal is longer. Yes, to digest cellulose for the herbivores, it takes a lot of time. The digestion process takes a lot of time. There are a lot of extra parts present in the herbivorous animals to digest the cellulose. That's why their intestines are little longer and more extra parts of the alimentary canal are present. But for frog, since it is carnivorous, mostly the diet of the adult frog is insect based. So protein hai, theek hai? So they are very healthy organisms. Mostly they are having protein diet. Okay. So frogs are carnivorous, their alimentary canal is short. That's why their intestine is also shorter. Now, a trick question. If they are asking tadpole, what you will say? Tadpole is carnivorous or tadpole is herbivorous? Adult frog is carnivorous, elementary length is canal short, intestine is reduced. Okay. What about the tadpole? Can you tell me quickly? Tadpole is also carnivorous, tadpole is also eating insects. Tell me fast. Bolo bhai, jaldi bolo. Tadpole is herbivorous. Tadpole is herbivorous. Tadpole is herbivorous. It is in the water. Chinna pilla yaar. Eating some algae and all, or some phytoplanktons and all. It is feeding on them. So it is basically a herbivore. Now don't ask sir, if it is herbivore, then the intestine will be long. Okay, don't go there. Okay, just remember herbivore. And then the remaining parts, mouth opens into the buccal cavity opens into the esophagus opens into the intestine. Okay? And two important glands are there with the digestive system, the liver and the pancreas. The two important glands are there, the liver and the pancreas. Are we clear? Okay? Moving ahead. Lastly, the intestine will open into the rectum and rectum opens into the cloaca. Liver secretes bile juice, which is a non-enzymatic juice, and stored in the gallbladder. Pancreas secretes the pancreatic juice, which contains the enzymes. Okay, okay. Diagram, see. We are having very little time, so some diagrams I may not revise, but remember that not a single diagram-based question came in the last paper. So all the diagrams are absolutely important. All the labelings of the diagrams are absolutely important. Not a single diagram should be taken casually. All the diagrams have to be seen carefully and their labelings also have to be seen carefully. Am I clear? Okay, moving ahead. Next. Food is captured by the bilobed tongue. Food is captured by the bilobed tongue. The tongue is mostly muscular, like your tongue is also muscular. A frog tongue is also muscular. It is written in the summary. You can check in the summary line quickly that the frog tongue is a muscular bilobed tongue. Okay. Digestion of the food starts in the stomach, I can say, where action of HCl on the food is there secreted by the gastric glands, the gastric juice, and then we are partially digesting the food in the stomach. That is called as the chyme. Okay, very important word. Partially digested food in the stomach, acidic food, chyme, that will move into the small intestine. Okay, and the first part of the small intestine, the duodenum is there, and duodenum is showing thrown into the folds. That is called as the villi. The duodenum is thrown into fold called as the villi. On the villi, there are columnar epithelial cells. On them, there is brush border. That is the microvilli. That will further increase the surface area for digestion and absorption. Now in the duodenum, which juices are being received? From the liver and the gallbladder, what is coming out? The bile is coming out. And from the pancreas, the pancreatic juice with the enzymes are coming out. That bile juice and pancreatic juice is coming into the duodenum via a common bile duct. Via which bile duct? Via a common bile duct. So gallbladder, let's say, gives one duct and the pancreas gives one duct. They both join and become a common bile duct that is opening in the duodenum. Bile is helping in emulsification of fat. In very simple language, what is emulsification here? Emulsification is basically breaking down the large lipid droplets into small chinna droplets. On them, the lipase enzyme of the pancreatic juice is going to act and digest them further. Okay, so basically breakdown. Okay, because the bile juice contain the bile salts, and bile salt kind of, they create the micelles. You might have studied that in chemistry, that micelles are created, the big droplets of the lipids are broken down into small droplets. Okay, chalo, moving further, the undigested waste will move out through the rectum and then into the cloaca. So first rectum and then outside the cloaca. Now talking about the next paragraph. Okay, what is this next paragraph is telling about? The next paragraph is telling about the respiration part. Okay, so before talking with respect to adult frog, tadpole respires via what? Come on, tell me. Tadpole respires via what? Come on, quickly in the chat box. Tadpole respires via what? Sir, I want to come tomorrow from the college offline. Is it necessary to listen the class? See, beta, it's upon you, yaar. Come on. If you feel you want to come offline, you are getting more serious here, you can come offline. If you think you can do with online revision, you can go ahead with online revision. Okay, it's not, no one is forcing you. Okay? Tadpole doesn't use a skin. Tadpole uses gills. Branchial respiration. Tadpole will use gills, branchial respiration. If you have not written, please write somewhere. Okay? Tadpole uses gills, branchial respiration. So I am telling some trick questions because little bit, if challenging question comes, basically these are easy questions. These are all covered in your long-term and the senior exams. All the different types of questions are being asked, but still you should be knowing. Now talking about the adult frog respiration. For adult frog respiration, first of all, the main is the skin. The cutaneous respiration, breathing via skin, cutaneous respiration. In the water, they are using skin respiration, that is cutaneous respiration. Then on the land, they can use skin, that is cutaneous respiration. They can also use the pulmonary respiration by their lungs, and they can also do the buccopharyngeal respiration. Buccopharyngeal respiration means basically they will keep their mouth open, and or even they inhale the air from the nostrils. As the air passes through the buccal cavity and pharynx, there are blood vessels which can help in the exchange of gases directly in the buccal cavity and pharynx also. So that is the buccopharyngeal respiration, we can say. So there is pulmonary, there is a skin cutaneous respiration, and there is buccopharyngeal respiration on the land. Only when they are in hibernating or estivating conditions, only when they are in the burrowing stage, that is hibernation or estivation. In hibernation or estivation, only and only skin cutaneous respiration will be used. Are we clear with this? And if I ask you out of all these three, which is the major respiratory mode? If I ask you which is the major respiratory mode, what you will answer? The major respiratory mode will be which one? Tell me. The major respiratory mode is the skin cutaneous respiration. ठीक है? Come on. Let's go ahead. Next. Now vascular system. ठीक है? We are done here. Okay, little bit more extra information if required. Actually, it's not required, but still I am saying frog is a negative pressure breather or a positive pressure breather, because today breathing topic is going to be happen. We are going to study that humans are doing negative pressure breathing. We create a negative pressure and then we inhale the air. What frogs are doing? Are frogs negative pressure breather or frogs are positive pressure breather? Come on, tell me. Frogs are positive pressure breather. Frogs are positive pressure breather. They cannot create negative pressure. They don't have the diaphragm. They don't have the intercostal muscles and all. So they cannot create a negative pressure. They are basically positive pressure breathers. So basically they will inhale air through their nostrils. They will close their mouth and they will pump the air inside. So they are positive pressure breather. We are negative pressure breathers. Are we clear with this? Remember this point. Moving ahead. Now we are going to talk about the circulatory system. Come on, circulatory system. It's a closed circulatory system. Frogs have well developed circulatory and lymphatic system, involves heart, blood vessels, etc. Lymphatic system consists of lymph channels and lymph node. Heart is muscular structure. Heart is a muscular structure. How many chambers heart? 3 chambered heart, 2 atria, 1 ventricle. What is the circulation? What is the circulation? Is it double circulation or incomplete double circulation? Tell me the answer quickly. Is it a complete circulation or is it an incomplete double circulation? It is incomplete double circulation. Incomplete double circulation. In the summary, again there is a mistake. In summary, there is a genuine mistake here that they have written single circulation. So again, I am telling this. In the summary, there is a small mistake. They have written single circulation from the frog. Please cut that. Please cut that. That is a wrong typo mistake happened. So that is an incomplete double circulation. Okay. Is frog heart myogenic or neurogenic? However, it is not written here. Frog heart is myogenic or neurogenic. Do they generate their beat on their own? The cardiac muscle generate the beat on their own, or they require neural input. Their heart is myogenic, like us. Okay, like we have the SA node, the pacemaker, they also have the SA node or the pacemaker, which is able to generate the regulatory impulses, which are helping in contraction and relaxation or stimulating the heart. Are we clear? Myogenic heart. Myogenic heart. Okay, this point is not written. Somewhere you can write myogenic heart is there. Next, moving ahead. Pericardium is there. We also have the pericardium. Okay, now the two important structure. One is the sinus venosus and another one is the conus arteriosus. So if this fist is the heart of the frog, this is the ventral and the backside will be dorsal. This will be the ventral part, backside will be the dorsal. What structure is in the ventral? What structure is in the ventral? Conus arteriosus. What structure is in the dorsal side? Dorsal side, there will be sinus venosus. So first remember what is ventral, what is dorsal. In the front, there is conus arteriosus. In the backside, dorsal side, there is sinus venosus. Okay, sinus venosus. On the dorsal side, there is a sinus venosus. What is the shape of sinus venosus? Sinus venosus is a triangular structure. Sinus venosus is a triangular structure which opens on the dorsal side of the frog heart and opens into the right atrium. Sinus venosus, triangular structure on the dorsal side, opens into the right atrium and it is collecting the blood from the major veins. Okay, three major veins are there which are opening in the sinus venosus. How many veins? Three major veins, or vena cava also you can say, that is opening in the sinus venosus triangular structure. In front, there is conus arteriosus. Conus arteriosus, what is the shape? Tell me. Conus arteriosus in the ventral side of the heart. What is the shape? Sac-like structure. What is the structure? Sac-like structure that is receiving the blood from the ventricles. That is receiving the blood from the ventricles. Okay, answer me. Is the ventricle pumping oxygenated blood, deoxygenated blood, or mixed blood? Is the ventricle pumping oxygenated blood, mixed blood, or deoxygenated blood? Is the ventricle pumping oxygenated, deoxygenated, or mixed blood? It is pumping mixed blood. Yes. It is pumping the mixed blood. So basically we have two atrias, right atrium receiving the deoxygenated blood, left atrium receiving the oxygenated blood from the skin and the lungs, and in the ventricle the blood is getting mixed. So the common ventricle, single common ventricle is pumping the blood in the sac-like conus arteriosus in the body. So the mixed blood is going there. So we are saying there is an incomplete double circulation. Are we clear with this? This I feel is a very important question. So sinus venosus and conus arteriosus, little be careful. A very important question. On this we can get some question. Okay. Next. Moving ahead. Going ahead now, special venous connection. Special venous connection, the portal circulation. In the frog, we are talking here two portal circulations mentioned. One is the hepatic portal circulation, another is renal portal circulation. What is the purpose of portal circulation? Okay, I know you know what is hepatic portal circulation. The blood from the intestine is going to the liver via the hepatic portal vein. It is the hepatic portal system, and from the lower blood, from the lower body of the frog, the blood is coming up and going to the kidneys. So that is the renal portal vein, collecting the blood from the lower body, going to the kidneys. Okay, you know this definition very well. But what is the purpose of portal circulation? What is the purpose of portal circulation? What is the purpose of portal circulation? Tell me. What is the purpose? Sir, it is just mentioned there. To drop some nutrients? What is the purpose, guys? What is the purpose? The purpose is actually to reduce the load on the heart. The purpose is to reduce the load on the heart. Imagine you are going from, let's say you are going from Vijayawada to Nagpur. Okay, and now Hyderabad is coming in between. Okay, so there are two roads. One road is going through the city and other is going through the bypass road. Which road you are going to take to go for a forward, a quick move out of the city? You are going to take the bypass road. So you are not getting congested in the traffic of the Hyderabad. Yes or no? Or the city. So basically what we are doing, we are creating a circulation. We are creating a circulation which is going to reduce the load of the heart. And that is a more sensible circulation. So what is happening in the intestine? Let's say you are absorbing all the nutrients. Again, it will go to the inferior vena cava. Again, it will go to the heart. And then again, it will be pumped to the liver. Liver will say, directly give it to me, na yaar. So what is happening? Intestine, spleen, stomach, small intestine, all the veins are getting joined and it is becoming a common hepatic portal vein, and that is going to the liver. So we are creating a shortcut. We are creating a shortcut and we are avoiding the long cut. And since we are avoiding the long cut, you are putting less load on the heart. Okay. Similarly, in the same angle, lower body, whatever the waste is generated, we are directly collecting it and passing to the kidneys. Kidneys will remove whatever the waste can be removed, and again, it will go into the body circulation. That is the use of the portal circulation. Okay, moving ahead, after renal and hepatic portal system, out of these two, which is present in humans? In humans, we have the hepatic portal system. We don't have the renal portal circulation. Along with this, we have one more portal circulation, that is the hypothalamo-hypophyseal portal circulation. Hypophyseal portal circulation is there. Hypothalamus releasing and inhibiting hormones which passes through the blood vessels and then reaches your anterior pituitary. That is the hypophyseal portal circulation. That is also there in humans. Okay, talking about the blood is composed of plasma and cells. RBC is there. RBC is nucleated here. Okay, underline the point, nucleated. We are enucleated. And it also shows hemoglobin, WBC, platelet is there. Lymph is different from blood. Same you can talk about humans also. Human lymph also is whitish in color. It lacks a lot of proteins. Okay, it lacks few proteins and RBCs. The blood carries, okay, this function we already know. Now we are directly jumping to the excretory part. We are directly jumping to the excretory part. So what is the excretory system made up of? Excretory system is made up of kidney, ureterus, cloaca and urinary bladder. There is a pair of kidney, there is a pair of ureter, single cloaca, single urinary bladder. Kidneys are compact, dark red bean structure. Okay, where they are situated? They are situated anteriorly or posteriorly? They are situated posteriorly in the body cavity on both sides of the vertebral column. Same for humans also. Each kidney, it is composed of the structural and functional unit, uriniferous tubules. Another name of nephron they have given, uriniferous tubules. Now, two ureters emerge from the kidney in the male frog. The ureter act as urinogenital duct which opens into cloaca. Very important. There is a functional, basically there is a functional connection of the male reproductive system and the excretory system of the male frog. Yes or no? That is absent in the female frog. Because whatever the sperms are made in the testes of the male frog, they are passing from the testes to the vasa efferentia. Vasa efferentia opens in the kidneys and then passes through the Bidder's canal of the kidney, which opens into the urinogenital duct. That is nothing but the ureter. So ureter of the male frog, we are calling it as urinogenital duct because major of the times it is conducting urine, but during the reproductive season, it can also pass the sperms. So that's why we are saying it is a urinogenital duct. For humans, what is the urinogenital duct? For humans, what is the urinogenital duct? Mukesh Khanna, you can revise tissue from the old NCERT, dear. The tissue part you can revise from the old NCERT, epithelial and connective tissue. That is little easier. Okay. For humans, it is urethra. For humans, it is urethra, the urinogenital duct, because urethra passes urine and also passes the semen containing the sperms. Okay. In females, ureters and oviduct open separately. See, in females, the ureter and oviduct open separately in the cloaca. Are we clear? So there is no connection between the ureter and the oviduct here. They are completely separate. Then there is a thin-walled urinary bladder. Now, what is the location of urinary bladder? Is urinary bladder in the front of the body or backside of the body? Urinary bladder in the front or the backside? Will it be ventral or will it be dorsal? What we will say? It will be ventral in position. In our also, it is ventral only. Okay, dorsally will be rectum. And the wall of the urinary bladder is thin. Wall of the urinary bladder is thin. Okay, hardly they are storing any urine. The urine capacity is also very less. For humans, the urinary bladder will be very thick. The detrusor muscle of humans, the urinary bladder is very thick. For frog, it will be very thin. Okay, present on the ventral side to the rectum and opens into the cloaca. Okay, frog is a ureotelic organism. The major waste excreted by frog is urea. Again, that is also a conservation mode of water to produce urea and also to produce a moderately toxic waste. What about tadpole? I will say. For tadpole, what I will say? Tadpole also I will say ureotelic only. For tadpole, what I will say? Tell me quickly. For tadpole, what I will say? Tadpole के लिए क्या बोलेंगे? Tadpole, it will be ammonia. For tadpole, it will be ammonia as the waste. So for tadpole, it will be ammonotelic. Are we clear, everyone? ठीक है? So if you have not written, just write somewhere. Okay? Excretory waste are carried by the blood into the kidney where it is separated and excreted. Okay, one generic question, everyone might be knowing this. Kidney are removing the waste. Who is producing the nitrogenous waste in frog? Which organ produces the nitrogenous waste in the frog? Which organ produces the nitrogenous waste in the frog? Or humans also? Yes, liver. Liver. Who produces the nitrogenous waste? The answer is liver. Are we clear? Liver produces the nitrogenous waste and it is removed by the kidneys. Okay, so keep your common sense always on. Okay, next, the system for control and coordination is highly involved in frog. Neural system, endocrine system, very well evolved. Okay, they also secrete a lot of hormones. Different endocrine glands: pituitary, thyroid, parathyroid, thymus, pineal body, pancreatic, adrenal and gonads are there, which are secreting the hormones. Nervous system is also well organized. Central nervous system containing brain and spinal cord. Peripheral nervous system contain cranial nerves and spinal nerves. Now, how many cranial nerves in frogs are there? How many cranial nerves in frog? And how many cranial nerves in humans? Peripheral nervous system, we are saying cranial nerve and spinal nerves. How many cranial nerves in frog and how many cranial nerves in humans? Humans have 12 pair of cranial nerves. Frog have 10 pair of cranial nerves. Humans have 12 pair of cranial nerves. Frog have 10 pair. Acha, what about spinal nerves? What about spinal nerves? Spinal nerves how many? Spinal nerves how many in frog? Spinal nerve also 10 pairs. Spinal nerves also 10 pairs. Spinal nerves in frog is also 10 pairs. So very easy. 10 pair of cranial nerve, 10 pair of spinal nerves. How many spinal nerve pair in humans? How many spinal nerve pair in humans? Humans have 31 pair of spinal nerves. Humans have 31 pair of spinal nerves. Okay? So frog, 10 pair of cranial nerve, 10 pair of spinal nerve. Whereas for human, you will write 12 pair of cranial nerves and 31 pair of spinal nerves. Okay? Now you can do the calculation. Are they asking in pair or are they asking nerves? You can double the number. Okay? So be careful there. Okay, next, autoimmune system, autonomic nervous system, that is sympathetic and parasympathetic also there in frog. So there are 10 pair of cranial nerves arising from the brain. Okay, brain is protected, like us also, brain is protected by the cranium. Okay, brain is divided into three parts: forebrain, midbrain and hindbrain. What is the forebrain made up of? The forebrain is made up of the olfactory lobes, which are primarily responsible for sense of smell. Okay, what is the forebrain made up of? Forebrain includes olfactory lobes. Now answer my question. Do human brain also have olfactory lobes? Forebrain includes olfactory lobes. Answer my question. Do humans also have olfactory lobes? Quickly, tell me the answer fast. Yes. Yes. It is not given in that nervous coordination chapter. It is not given in the nervous coordination chapter directly. They have not mentioned. They forgot to mention actually. Okay. So in humans also there is olfactory lobes. They forgot to mention in the forebrain part in nervous coordination chapter, but remember this. Olfactory lobes for sense of smell. For us, it is more developed as compared to frogs. Are we clear? So olfactory lobe is there. ठीक है. Paired cerebral hemisphere, that also we have. Unpaired diencephalon. Unpaired diencephalon. We also have diencephalon. Frogs also have diencephalon. What is basically diencephalon? Diencephalon is nothing but thalamus. You should also know the common name of diencephalon. Diencephalon is nothing but thalamus. Are we clear? Yes. In human forebrain also olfactory lobes are there. Olfactory lobes are in fact associated with limbic system also. So now if you go and study the nervous coordination chapter summary part, in summary part of the nervous coordination chapter, you can read in the paragraph, they have mentioned the limbic system, olfaction function they have mentioned. Olfaction is sense of smell, and that is associated with the olfactory lobes. Are we clear? Just below the cerebrum, the olfactory lobes are present. So you have the two big cerebral hemispheres. Just below them, there is the olfactory lobes. From them, the olfactory nerves are coming in your nose. From olfactory lobes, the olfactory nerves are coming in your nose, which are responsible for the sense of smell. Are we clear? Location of diencephalon, cerebral hemisphere. Below cerebral hemisphere, we are having the diencephalon or the thalamus. Are we clear? Are we clear, guys? Shall we go ahead? Don't worry, yaar. When we talk about location of diencephalon, below the cerebrum, yaar, come on. Below the cerebrum, there is thalamus. Okay? So when we go to nervous coordination, again, I will tell you to go and visit. Few more points are going to come shortly. Listen. Midbrain. Midbrain is characterized by a pair of optic lobes. How many lobes in us? Frog midbrain consists of two optic lobes. Optic means visual. So basically, these optic lobes are majorly associated with the visual function, the visual inputs or the visual reflex. For us in midbrain, how many lobes are there? We have 4 lobes. Corpora, quadrigemina. Frog has 2 lobes in the midbrain, 2 optic lobes. That is a pair of optic lobes. I can say corpora bigemina. Corpora bigemina in frog. For us, it will be corpora quadrigemina. Our upper 2 lobes are optic lobes. Lower 2 lobes are actually the auditory lobes. Superior colliculi, inferior colliculi. Superior colliculi in humans for visual inputs, visual reflex. Lower 2 colliculi are responsible for the auditory inputs, auditory reflex. And also the midbrain is responsible for the tactile inputs, touch input. So again, if you can go and see the nervous coordination summary part, there they have mentioned the midbrain function, the midbrain corpora quadrigemina, the superior colliculi, inferior colliculi, visual, auditory and tactile inputs they are associated with. For frog, midbrain, it is only how many lobes? A pair of optic lobe that is doing just the visual function. Are we clear with this, guys? Are we clear? See, guys, why I am telling you this point associated with brain? Because if I am the examiner, I will combine those points and make the question. So if such examiner comes, I pray to God that such examiner will not come, but I'm still trying you to relate that points. Are we clear? Sir, is corpora quad or bi? For frog, it is bigemina. For human, it is quadrigemina. For frog, it is bigemina. For human, it will be quadrigemina. Are we clear? Chale, theek hai? Yes? Then the medulla oblongata passes out through the foramen magnum. The medulla oblongata passes out through the foramen magnum. What is foramen magnum? Foramen means a big opening. Okay? Magnum, big size. Foramen is opening, magnum is big size. So basically on the occipital part of the cranium here, there is a big opening, that is the foramen magnum, through which the medulla oblongata comes out and extends as spinal cord. We also have the foramen magnum in our also occipital bone here. We have the opening, foramen magnum, through which the spinal cord is going to pass out. Now, frog, they are telling here, there are 5 sensory organs in the frog. Sense of touch, sense of vision, sense of smell, sense of hearing, sense of taste. Out of all these 5 senses, which are the most developed ones? Which sensory organs are more developed in the frogs? Which sensory organs are more developed in the frog? Sense of vision, sense of hearing. Sense of vision, sense of hearing. Okay? Yes, Mukesh Khanna. Auditory function is done by the tympanum. Yes, correct. Remaining all are C. So frog has different type of sense organs. Organ of touch, sense of touch, sensory papillae, taste buds, smell, nasal epithelium, vision, eyes, and hearing, tympanum with the internal ears. Outside tympanum connected inside with the internal ear. Out of this, eyes and internal ears are well organized structure. So can be asked as an MCQ, which of the following sensory structure of the frogs are well developed? So that is the eyes and the internal ears. Rest are cellular aggregations. Remaining are just neurosensory epithelial cells. Neurosensory epithelial cells means On the tongue, we have taste buds. They are epithelial cells connected with sensory nerve, and the nerves are supplying to the brain. So whatever you eat, whatever taste you get from that epithelial cell connected to the nerve, sensory neuron, that is going to your brain. Similarly, the sense of touch. Similarly, the sense of smell also. Are we clear? Next, ear is an organ for hearing and balancing as well. For us also, ear is an organ for hearing and balancing both. Now talking about the reproductive system. Male reproductive organ consists of a pair of yellowish ovoid testes. A pair of yellowish ovoid testes present on the kidneys. Present on what? Present adhered to the upper part of the kidney by double fold of peritoneum called as mesoorchium. This line is very important. Double fold of peritoneum, mesoorchium. So basically mesoorchium is a kind of a membranous fold which is holding the testes on the upper part of the kidneys. Okay. From the testes, what is coming out? Testes are containing seminiferous tubule that are producing the sperms, and from that testes, vasa efferentia ducts are coming out. What are coming out? Vasa efferentia are coming out, and vasa efferentia, they are how many in number? They are 10 to 12 in number. How many in number? 10 to 12. Do human testes also have vasa efferentia? Do human testes also have vasa efferentia? Tell me. Do human testes also have vasa efferentia? Tell me fast. And if they are there, human testes also vasa efferentia is there. So you tell me, for frog it is 10 to 12. We will have more or less. We will have more or less. Vasa efferentia in frog is 10 to 12. Will humans have more or less? Humans have more. Human have roughly 12 to 20 vasa efferentia ducts. 12 to 20 vasa efferentia ducts. Humans have little more. Humans have 12 to 20 vasa efferentia ducts coming out. Okay, so someone is asking some questions, sir, what are the fat bodies? What are the fat bodies? Fat bodies are fat bodies. Or fat bodies are fat bodies. They are kind of an adipose tissue. They are storing some fat. So during hibernation and estivation, that will be used as a source of energy. Okay. So during their hiding stage, hibernation and estivation, when their body metabolism is very low, that energy is provided by the fats. Are we clear? And since fat is very close to the testes, fat bodies are very close to the testes, some fat deposition surrounding the testes also happened. So when you look the testes from outside, they are going to give a yellowish appearance because fat also gives a yellowish appearance. So that's why we say yellowish ovoid bodies. So you can see these are the testes. From testes we can see these are the ducts coming out, the vasa efferentia. They are opening in the kidney. This is the Bidus canal, and from the Bidus canal they are passing into the ureter, and ureter in the male frog will be called as the urinogenital duct. What is the function of vasa efferentia? Vasa efferentia is basically a duct. Vasa means a duct. Efferentia means going out. Afferent coming in, efferent going out. Efferent is going out. So vasa efferentia, a duct, something is taking out from the testes, that it is taking out the sperms. And then it comes in the kidney. As it comes in the kidney, you can see these are the horizontal canals and the vertical canals. These are the Bidus canal, ultimately connecting to the urinogenital duct. Okay. Moving ahead. Moving ahead. Come on. Finally, so this question many times asked in the exam. Many times, past also 2-3 times the PYQ has came, the pathway of the sperms from the testes to vasa efferentia, then kidney, then Bidus canal, and then the urinogenital duct, and then from the urinogenital duct, the cloaca, then it will be exiting out many times. Okay, okay. Next, the cloaca is a small median chamber that passes the fecal matter, urine, sperm, etc. to the exterior. Correct. Then female reproductive system consists of a pair of ovaries. The ovaries are situated near the kidneys and there is no functional connection. Very important line. There is no functional connection. Like here the testes have the vasa efferentia opening in the kidneys and there is a Bidus canal which is kind of a functional connection between the testes and the kidneys because Bidus canal are on the kidneys. Okay, so there is no functional connection here with the kidneys. A pair of oviduct arising from the ovary. Okay, oviduct separately opens into the cloaca and at a time they can lay about 2,500 to 3,000 eggs. Fertilization is external inside the water. So basically they are doing spawning. You might have heard the word spawning. Mostly whichever organisms go for external fertilization, they are releasing their gamete outside in the water. So that is the spawning release of the gametes at the same time in the water. External fertilization, indirect development, larval stage. The larval stage is called as tadpole. Tadpole will metamorphose into the adult. Are we clear? Yes? Sir, statocysts are balancing organ. Statocysts are the balancing organ we studied in aquatic arthropods. In aquatic arthropods. And spawning word, yes, it came in cyclostome. Very well. Sir, vasa efferentia is intratesticular or extra? Vasa efferentia is intra. Vasa efferentia arises from the testes, from inside. So it is intratesticular. Vasa efferentia arises within the testes and goes outside. Since it is arising from the testes and then going outside, where it started from? It started from the inside. So it is intratesticular. This question was asked in human reproduction system also we ask that seminiferous tubule, retetestes and vasa efferentia intratesticular. Epididymis, vas differentia, they are outside completely the testes. Then it becomes the extratesticular. Sir, will you say the pathway of the male gamete? Come on. Testes. From testes, it will go to the vasa efferentia. From vasa efferentia, it will go to the kidney. From kidney, it will go to the Bidder's canal. From Bidder's canal, it will go into the urinogenital duct, that is ureter. And from ureter, it will go to the cloaca. Okay. Definitely, if we are asking this paper, this question will be definitely coming. Because many times, this is the most commonly asked question. Are we clear? Chalo, let us continue. This paragraph I am not going to read. Okay. This you have to read on your own. Okay. Anything extra given in the summary points? No. Nothing given, anything extra. Okay. Now directly moving to the next chapter. Let us start with breathing chapter. Come on. Shall we start with breathing chapter? Come on. No. Frog do not use fat body for excretion role. That was cockroach. Cockroach, they little bit store the uric acid in the fat bodies. So fat bodies had a role in excretion there in cockroach, not for frog. Okay. You are connecting the two topics. You are connecting the fat body with the with the cockroach topic. Come on, now the next topic, breathing and exchange of gases. Very good question asked by Catherine. Catherine is asking, sir, metamorphosis is done by which hormone in frog? Come on, everyone tell me the answer. Metamorphosis in the frog is done by which hormone? Come on, which hormone? Present here. In humans, the gland is present here. Thyroid gland. Yes. अरे यार सैयद हाइंड लिम फोर लिम कौन सा खा सकते हैं? दोनों खा सकते हो, but hind limb is more muscular, so you are eating like chicken leg piece like you eat. So frog also we eat the leg piece only. We means other people who eat it. So the frog hind limbs are used as a food delicacy. Correct answer. Frog metamorphosis is definitely done by the thyroxine hormone, but obvious iodine is also important, because without iodine we cannot make the thyroxine hormone. Come on, now let's go to the breathing chapter. Come on, quickly. So before going to the breathing chapter, this is a paragraph given here. Human physiology. ठीक है? Human physiology. First you will read this paragraph, okay? The reductionist approach. 2025 question came from this. What is the reductionist approach? Physiochemical approach. You study the physical, chemical, both the aspects, to study how the cell or the organ systems are working. So you have to revise this, okay? You have to also revise all the scientist which is given. Don't skip any scientist, Okay, all the scientists, whatever is there after any section which is starting, like in the ecology section, we had that Mishra person, that was the father of ecology and all. Similarly, here also there is mentioned Alfonso Corti. Alfonso Corti, okay, birth date, that date they will not ask mostly. Mostly they will be asking what they studied and why, what special study they did. Like this guy, Alfonso Corti, started with the studying of cardiovascular system, and then he suddenly went to the internal ear and he discovered the organ of Corti. So he discovered a structure which is responsible for hearing, a sensory structure which is responsible for hearing, the main organ of hearing inside the internal ear. So it was named on his name, organ of Corti. Are we clear? Basically, organ of Corti consists of basilar membrane in the cochlea, right? That helps to convert the sound vibration into the nerve impulses. Okay? Are bhai dekho. Lot of things are not asked in this paper. Like scientists, they have not asked, so they might ask. They didn't ask any assertion reason. Hello. Assertion reason, this time it will be there now. But again, I'm not scaring you. Whatever assertion reason questions are going to come, they are coming directly from the NCERT lines. Okay, they will be related to NCERT lines. Okay, so we were lucky to not have assertion reason last time, but again, we were prepared for it. Now also we are prepared for it. Okay, so if it is very tricky, it is tricky for everyone. And mostly they are giving straightforward assertion reasons they are giving. Either the reason will explain or the reason will not explain. Okay, they are not confusing you. Straightforward reasons, whichever lines are explaining the NCERT assertions, that type of questions they are going to ask. Are we clear? ठीक है? So don't create a panic that, okay, already that, sir, they are going to ask a lot of difficult assertion reason questions. No, there are 3 or 4 good assertion reason statements naturally present in every chapter of zoology. So from that, they can pick any lines which can be related as assertion reason questions. Are we clear? Sir, what is the relation of fat with temperature? Arey, fat with temperature means if adipose tissue is more, then you are going to being protected from the temperature. Na? Fat helps you to protect against the cold temperature. Okay, and in cold temperature, if you are not going to get anything to eat, then fat will also act like an energy source. It will also going to give you energy. ठीक है? So system biology in introduction. Okay, everyone is asking this system biology. System biology. Arey kya bhai? What is the meaning of system biology? We are studying which system? Respiratory system. Then we will study excretory system, nervous system. What is this? System biology. So how a particular system, organ system functions is system biology. Now, an organ system is made up of multiple organs. Yes or no? Like we are going to study breathing system, respiratory system. That is made up of lungs, hai, trachea, hai. Yes or no? Nose is also involved. Okay? So many parts of respiratory systems are there. How every system is working? What is the benefit of the conducting system to the respiratory system? You are saying, sir, conducting system is able to remove the foreign particles, remove the dust and everything, humidify, bring it to the body temperature. And then you say, sir, what is the function of the incomplete cartilaginous rings on that trachea, primary bronchi, secondary bronchi? So they are preventing the collapsing of the tubes so that the air pathway is completely opened. Yes. So see, whenever we want to study any system, we have to have an approach to study into in small parts. First, understand from basic. And then as you understand basic, you will understand how the whole system works. Like we always don't jump directly to system. First you study cell, then you study tissue, then you study one small organism, and then you study that small organism's functioning. Then you go to human system, and then you talk about human organ system, different parts. That is the understanding of system biology, and how we have understood the system biology, that is mentioned here. Okay, so what they are writing here, see, system biology makes us believe that all living phenomena are emergent properties due to interaction among components of system under study. Now simple. In the system biology, let's write digestive system biology. Digestive system biology makes us believe that all living phenomena, that every living organisms which is digesting their food are emergent properties due to interaction. Why that digestion is happening? Due to the property of different organs involved in that system. Are emergent properties due to interaction among the components of the system. Like if stomach is digesting the food, making it acidic, making it chyme and soft, and then it goes into the intestine, then there it is made alkaline, and then you are doing the enzymatic digestion further and completing the digestion process, and then it is being absorbed. Is it connected or not? Like if you don't chew the food properly, the digestion itself starts in the mouth where you are chewing the food and digesting. So basically the mouth is also interacting in the digestive system. It is doing the physical digestion. So if some person which is not having teeth, toothless guy, not able to chew the food properly, do you think not chewing the food properly is going to make the stomach upset? Yes. Are we clear with this? So this is what they are telling. So system biology is helping you to understand its function, how the different components are interacting, how the different components of the systems are interacting. Now to study that regulatory network of molecules, supramolecular assembly, cell, tissue, organism, indeed population and community. Now what this line is telling you, first you study cell, then you go for the tissue, then you go for the organ, then you go for the organ system. And when you understand an organism, then you focus on the group of the organism. Group of the organism is population. Then there are different population. Then you focus on how population are interacting. That is community. Where is the community present? The community is present in the biome. Then you start studying the biome. So suddenly you start from a cell. Then you go to the physiology. That is the system biology. Then you go for the organism understanding. Then you go with the ecology. Are you getting me, guys? Yes. Like this studying system biology, you have discovered a lot of human physiological system and their understanding we have developed. Are we clear? Yes. Yes, NTA has announced 21st June. Chairman sir gave a letter to NTA to conduct on 21st June. So it is conducting now. Okay. Shall we go ahead? Now, this paragraph, remember the definition of breathing. What is breathing? Indirect breakdown. Okay, remember this word. Oxygen is utilized indirectly. We are not utilizing oxygen directly. If you utilize oxygen directly, there will be explosion of energy. That's why we are utilizing the oxygen indirectly. There is a stepwise way of producing energy or ATP. First you do glycolysis, then you convert to pyruvic acid, then you send into the link reaction, and then you send it to the Krebs cycle, and then you generate the NADH2, FADH2. Some direct ATPs are generated in between, and then you channelize into the, the ETS, where you are doing the proton pumping. And then after that, you are able to synthesize the ATP. So that is an indirect use. In the last step, the oxygen will be coming. That's why we are saying indirect breakdown. Okay, come on. Respiratory organs, I will not revise this, you have to go through this. Initially they have mentioned invertebrates, sponges, coelentrates, flatworms. They are using directly their body for surface for exchange. Earthworm, moist cuticle. Insects, they will use trachea. Which insects will use trachea? Terrestrial insects will use trachea, whereas aquatic arthropods, the arthropods which are in the water or the mollusk, aquatic arthropod or the mollusk which are present in water, they will breathe via gills, and gills respiration is called as branchial respiration. Whereas mollusk which are present on the land, they will go for what? They will go for what? They will go for breathing via lungs. Next, among vertebrates, fishes use gills. Amphibians, they use everything amphibian we have studied. They can breathe via gills also if they are in the tadpole stage. Majorly they use cutaneous, pulmonary and skin. Mostly cutaneous, moist skin, cutaneous, and also their lungs. Reptile birds and mammals, definitely reptile birds and mammals, they all are using lungs. Okay, moving ahead. Next is the Human respiratory part, again, I will not go through all this. I will tell you the most important part. The most important part in this paragraph, you have to remember this line. Trachea is straight tube extending up to mid-thoracic cavity, okay, where it divides at the level of fifth thoracic vertebra. So this is a very anticipatory question, okay, fifth thoracic vertebra. Then the most important thing as per my thinking is that in this paragraph, which are lined by incomplete cartilaginous rings and which are not lined. So tell me which tubes are having incomplete cartilaginous rings and which are not having them. Trachea, primary bronchi, secondary bronchi, tertiary bronchi, initial bronchioles. Yes, till initial bronchioles, we will say the incomplete cartilaginous, C-shaped cartilaginous rings are present to prevent the collapsing of the lung, to prevent the collapsing of the tubes. Whereas it is not present where it is not present, it is not present on terminal bronchioles. It is not present on the terminal bronchioles. Okay? It is not present on terminal bronchioles. It is not present on the respiratory bronchioles. It is not present on the alveolar ducts. It is not present on the alveoli. So, starting from the trachea till initial bronchiole, someone in the YouTube said terminal bronchiole also has it. No, that is a mistake. Okay, so just read that line. The trachea, primary, secondary, tertiary, and the initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchioles give rise to very thin, irregular walled vascularized alveolar structure. See, after initial bronchiole, there will be terminal. After terminal, there will be respiratory bronchiole. Respiratory bronchiole is not mentioned here, but after respiratory bronchiole, there will be alveoli ducts and then the alveoli, which are not having the rings on them. Next, alveola is protected by a double membranous pleura, outer parietal pleura, inner visceral pleura. What is the pleural membranes are having between them? The pleural membranes are having what between them? The pleural membranes are having the pleural fluid. What is the function of pleural fluid? It prevents the friction. It prevents the friction. That's all? No. One important point you will write down, that the pleural pressure is a negative pressure. Interpleural pressure is a negative pressure. Come on, somewhere note down. On your textbook only note down. Interpleural pressure is a negative pressure which also prevents the collapsing of the lungs. Interpleural pressure. Interpleural pressure. Interpleural pressure is which pressure? Interpleural pressure is a negative pressure which prevents the collapsing. Now don't get scared. Don't get scared. This question was asked in the PYQ. This question was asked in the PYQ. That's why I'm telling this point. So in the pleura, the outer parietal and inner visceral, there is a negative pressure. Okay? Negative pressure means roughly minus 5 mm of Hg, the pressure is there. And as you inspire, the further negative pressure might increase. That negative pressure is preventing the collapsing of the lung. Are we clear? That's why one question was asked, if some object punctures your pleura but doesn't puncture your lungs, some sharp object, if it punctures your lung, then you are dead only. If it punctures the lung, we are dead. Because if the lung puncturing occurs, all the air will rush inside. Then you will not be able to breathe. That airtight chamber, the anatomical setup is gone. Yes? But if it just punctures the pleura, if it just punctures the pleura, let's say it pierces the parietal pleura and little bit it goes inside, but doesn't touch the visceral pleura, not coming in contact with lungs, what will happen? From that puncture, the atmospheric air will rush in. Now in the pleura, which was having a negative pressure, kind of a vacuum it was there, a negative pressure was there. Now the air will go there. Now the air will rush. Now a positive pressure will be created, and that will put pressure on the lungs and lungs may collapse. And this condition is called as a pneumothorax. This condition is called as a pneumothorax. Why I'm telling you? Because if some extra things are asked, okay, you should be knowing this. So pneumothorax is an important concept where students have this mostly doubt in this pleural topic. Remaining else, we all know, yeah, pleura prevents friction. That's all. Okay. Are we clear with this? Are we clear with this? Negative pressure means the pressure which is lesser. Pressure lesser than atmospheric pressure will be called as the negative pressure. Okay. Pressure lesser than atmospheric pressure will be negative pressure. Are we clear? We also create negative pressure in the lungs also. We also create the negative pressure in the lungs also. Okay. Now, next line. Moving ahead again. All diagrams are important. Epiglottis, larynx, trachea, pleural fluid, cut-end. All the diagrams, every labeling you have to go through carefully. Next, moving to the next paragraph. Now this paragraph here. In this paragraph, the conducting part and the exchange part or the respiratory part you have to remember. Conducting part is from where to where? Conducting part is from the nostrils till where? Till where? Till terminal bronchioles. The conducting part from where to where? The part starting from external nostrils to terminal bronchioles is your conducting part. Then what is the function of conducting part? Removes the foreign particles, humidifies the air and brings to the body temperature. After terminal bronchioles, there is alveoli ducts and alveoli. They are the part involved in the exchange of gases. So that is the exchange part, correct? Whereas the alveoli and their ducts form the respiratory or exchange part. So very important, conducting part or respiratory or exchange part. Question can be coming from this paragraph as well. Next is the anatomical setup. The anatomical setup they are talking about. Your lungs are present in a thoracic cage, a thoracic chamber. What are the boundaries? Anteriorly, ventrally, we have sternum. Dorsally, we have vertebral column. Laterally, we have the ribs. And on the lower side, we have the dome-shaped diaphragm. And this thoracic cage is an airtight chamber. This anatomical setup is an airtight chamber. Okay? And then, why this airtight chamber is important? The anatomical setup of lungs in thorax is such that any change in the volume of thoracic chamber will be reflected in the lungs or the pulmonary cavity. That's how you are able to inhale and exhale by indirectly manipulating the volume of the thoracic chamber and then manipulating the pulmonary volumes and pulmonary pressure, because you cannot do it directly. Can you directly pump air and suck out air? No, you cannot do it directly. You are doing it indirectly. So such an arrangement is essential for breathing, as we cannot directly alter the pulmonary volume. We are doing it indirectly. So we are negative pressure breathers. We are creating a negative pressure. What is a negative pressure? A negative pressure is a pressure which is less than the atmospheric pressure. So when in your lungs, inside the pulmonary cavity, when you create a negative pressure, then only you are able to inhale the air inside. The air will pass from the higher pressure to the lower pressure. Are we clear? How many lobes are there in right and left? So on the left side, we have the heart. So on the left lung, we will say 2 lobes. On the right side, we will say 3 lobes. Right side, 3 lobes. Left side, 2 lobes. Sir, what is the meaning of ventilation? Ventilation and breathing are same. Inhalation and exhalation of air is same as ventilation. So you can say breathing, respiration, ventilation, same. But when you're talking about cellular respiration, how the cell utilizes the oxygen, then that will be called as the cellular respiration. See here, breathing and pulmonary ventilation is the same thing. Okay, the physical process of inhaling and exhaling the air. Okay, when the cells are utilizing the oxygen for the breakdown process, then it will be called as the cellular process. Come on, now mechanism of breathing. Okay, let us start with mechanism of breathing, guys. Come on. Breathing involves 2 steps. One is inspiration, one is expiration. Which one is passive and which one is active? Tell me. Which one is a passive process? Which one is an active process? For inhalation, you are contracting your muscles, diaphragm and external intercostal. So it is an active process because you are using your muscles. Whereas for expiration, what you are doing? For exhalation, you are doing nothing. Exhalation, your diaphragm is relaxed. Your external intercostal muscles are relaxed. So here you are not doing anything. It is a passive process. So remember, it is not written here, but you have to write. Inspiration is an active process where you are contracting muscles using some energy. Whereas expiration is a passive process where you are relaxing the muscle. So you are exhaling the air without any effort, without any use of muscle contraction. Are we clear? So, and again, I am talking for normal breathing. I am talking for normal breathing. Because if you are doing forceful breathing, if you are doing forceful breathing, then you can say in exhalation also, you use certain muscles to exhale the air fast. Then I will say forceful breathing, exhalation, inhalation both becomes active. In forceful breathing, exhalation, inhalation both becomes active. But probably that concept, these NTA guys will not ask. Mostly inspiration active under normal conditions, normal resting conditions. And for exhalation, it will be passive. Now, to inhale air, we are requiring some respiratory muscles. We are requiring the respiratory muscles. So which are the two most important respiratory muscles we are using? We are using diaphragm and we are using the intercostal muscle. We are using the diaphragm and we are using the intercostal muscles. Very interestingly, diaphragm is also a skeletal muscle. Intercostal muscles are also skeletal muscle because both are connected to bones. Okay? Diaphragm and external intercostal both are skeletal muscles. However, their control is both voluntary and involuntary. Their control is both voluntary and involuntary, especially for diaphragm. So diaphragm is controlled both voluntary also and involuntarily also. So normally when you are sleeping, you are not paying attention to your diaphragm, but you are breathing normally. So that is involuntary. But when you are voluntarily breathing, like you are doing any yoga, you are monitoring your respiratory rate. When I tell you inhale, exhale, stop your breathing. So that is a voluntary act you are doing. Then it will become voluntary also. So diaphragm is both voluntary and involuntary, and it is a skeletal muscle. What it is? A skeletal muscle. Diaphragm normally is a dome shape. Diaphragm is normally dome shape under resting conditions. Okay? Whereas the muscle present between the ribs. So let's say these two fingers are the ribs and the muscle present on them outside and inside will be called as the intercostal muscles. The muscles which are present on the outside, external intercostal muscles. The muscles which are present on the inside, we will say the internal intercostal muscles. Now let us start with the breathing mechanism. So in inspiration, you want to inhale the air inside. In inspiration, you want to inhale the air inside. Outside pressure you cannot change. Outside pressure is the 1 atmospheric pressure. So what you have to do to take the air inside? To take the air inside, you have to create a negative pressure. It means you have to create a lesser pressure inside your lungs. Your intrapulmonary pressure should be negative as compared to the external outside pressure. So what we are going to do here? We are going to take the help of your rhythm center. The respiratory rhythm center in the medulla will send signal first to diaphragm. First, the signal will get to the diaphragm and then the diaphragm is going to contract. Diaphragm is normally dome shape. It gets contracted, it goes below. As the diaphragm is going down, it is becoming flattened. When the diaphragm becomes flattened, what is going to happen? The volume is increasing in vertical axis. The volume is increasing in vertical axis because diaphragm was dome, now it is flattened. So we will say the diaphragm contraction leads to diaphragm flattening, and that increases the thoracic volume in which axis? That increases the thoracic volume in anterior posterior axis. Okay? Then it is quickly followed by contraction of external intercostal muscles. The muscles which are present on the outside. External intercostal muscles will contract and then they will pull the rib cage outwards and upwards. So when we are inhaling, when we are inhaling air, what is happening? The rib cage is coming outwards and upwards. The rib cage is coming outward and upward. So when external intercostal muscles contract, rib cage comes outwards and upwards. That increases again the thoracic volume in the dorsoventral axis. In which axis? Dorsoventral axis. So these are the important lines. I am just saying the NCERT lines. Okay, you all know these points. So just I am recalling that. Diaphragm contraction first, gets flattened, increase in anterior posterior axis, external intercostal muscles contract, pull the rib cage outwards and upwards, dorsoventral axis thoracic volume has increased. So overall there is increase in thoracic volume. The thoracic chamber volume increases, and we know whenever volume increases, pressure decreases. So as the thoracic volume is increasing, what is happening to the pressure inside the lungs? The pressure inside the lungs is declining. The pressure is going down. So we say the intrapulmonary pressure is becoming negative. Boyle's law. volume increased, pressure decreased, pressure is decreasing, and the pressure is decreased so much that the atmospheric pressure becomes higher comparatively, relatively. Now the air is going to rush inside. Yes. So it happens simultaneously. So as your chest expands, thoracic chamber increase in volume, pressure declines in the lungs, and the air rushes in. It happens simultaneously. So for inspiration, what we did? We contracted the diaphragm, it became flattened, increased in dorsoventral, increased in the anterior posterior axis. We contracted the external intercostal muscles. We increased the ribcage expands, comes outwards and upwards, increase in anterior posterior axis, and we increase the thoracic volume, decline in the intrapulmonary pressure. That will lead to inhalation of the air. Now what happens? Now the signal coming from the medulla respiratory rhythm center, it stops. As the signal stops, diaphragm will again relax. External intercostal will also relax because the signal is stopped. Diaphragm will again go back to its resting dome shape. External intercostal muscle will release the rib cage. Rib cage is going to go downwards and inwards. Downwards and inwards. Your lungs are already filled with the air. Now suddenly what is happening, as the diaphragm is getting again back to dome shape and the rib cage is coming down and inwards, that creates what? That creates decrease in the thoracic volume. There is a decrease in thoracic volume due to which the pressure inside the lungs will increase. Already your lungs are filled with air, the pressure will increase and the air will rush out. Are we clear? So in expiration, what is going to happen? Diaphragm and external intercostal muscles relax. Okay? Due to which thoracic volume decreased, intrapulmonary pressure increased, air rushes outside. Here you did not contracted any muscle. So it is a passive process. Passive process will take a little bit more time. Okay? 3 seconds for exhalation under normal resting condition, 2 seconds for inspiration. 2 second inspiration, 3 second exhalation. In 1 minute, there is 60 seconds. So if 5 seconds for one breathing 5 seconds, 2 second inspiration, 3 second exhalation. So the minimum breathing rate you can get is 12 5 is 60. So 12 breathing per minute is the normal breathing rate. Maximum 12 to 16 we can tell is the normal breathing rate. Are we clear? So tell me, did we use internal intercostal muscles in normal breathing? Tell me, did we use internal intercostal muscles in normal breathing? In normal breathing, internal intercostal muscles are not used. Internal intercostal muscles are never used in normal breathing. In normal breathing, normal quiet breathing. See, when I say normal, normal is quiet breathing. You are right now quiet. Okay? In normal quiet resting breathing, you are not using internal intercostal muscles. Only during forceful exhalation, or when I am shouting, or I am singing, or I am producing voice, or when you are speaking, you have to forcefully throw the air through your vocal cords. Then only you will use internal intercostal. Internal intercostal is lying inside, so it will pull the rib cage inside and it will throw the voice out. So in that case, forceful breathing, we can say internal intercostal muscles are used. Mostly they are used for forceful exhalation. Are we clear with this? Are we clear with this? And you can also use one more thing. Don't forget the abs. Don't forget the abs. Abdominal breathing. Abdominal breathing. Okay. You also use your abs when you're doing forceful breathing. Okay. So in forceful breathing, you're also using abdominal muscles. You can also use the internal intercostal muscles. Are we clear with this, guys? Okay, again, both the diagrams are important. Labelings are important. Carefully go through the diagrams. Okay. Volumes and capacities, any doubt in this? Volumes and capacity, any doubts in volumes and capacities? Yeah? Any doubts in volumes and capacities? Tell me, anyone? Tidal volume is the amount of air inhaled or exhaled per breath. Amount of air inhale or exhale per breathe is 500 ml. Per minute, if it is there, you have to calculate with the breathing rate. So be careful if they are telling you to calculate the tidal volume. Like if you go and check the exercise question. Yes. Every chapter now you will see the exercise question. Every chapter you will read the summary. If you go and read the exercise question, in the exercise question you will read, in the exercise question you will find calculate the tidal volume per hour. Tidal volume per breathe is 500 ml. Okay. So if you take minimum breathing, 12 breathing per minute, so 500 into 12, whatever is the answer, and in an hour how many times you are going to breathe, you have to accordingly multiply and tell the answer. Okay, so be careful with that. Okay, are we clear? Sir, we are getting some sort of tension in the ribs. Is there any relation to intercostal muscles? No, that happens when you are doing exercise. You get certain pain in the abdomen here when you are doing certain exercise. Well, without warming up, if you do any exercise, suddenly the abdomen muscles, they pain because they are not used to do forceful breathing normally. Okay, so little bit pain may come and nearby the ribs are there. So that pain might occur. Are we clear? Moving ahead, volumes, okay, no tension in volumes. Exchange of gases. In exchange of gases here, three things are very important. The partial pressure gradient difference, the membrane thickness, the respiratory membrane thickness less than 1 millimeter, and then the solubility of the CO2. First is the partial pressure gradient. For oxygen exchange, we are having a very high partial pressure gradient. In the atmosphere, we are having 159. In the alveolar air, we are having 104. In deoxygenated blood, 40. In oxygenated blood, 95. In tissues, 40. So what I'm telling you here is, we have to by heart the values, no doubt. But you can see there is a lot of difference. There is a difference between 159 and 104. That difference helps in exchange of the gas. Again, 104 to 40. That difference also is there. A lot of gradient difference is there. Here also 40, 95, lot of gradient difference is there. Same between the tissues and oxygenated blood. So for oxygen diffusion, there is always a high partial pressure gradient difference. That is helping the oxygen to diffuse from the alveoli to blood capillary, from the blood capillary oxygenated to the tissues. Whereas if you notice for CO2 in our body, everywhere the partial pressure difference is hardly 5 mm of Hg. Like in tissues it is 45, in oxygenated it is 40. So from 45 it will go to 40. From deoxygenated blood it is 45, in alveoli it is 40. From 45 it will move to 40. And but obvious from alveoli 40 it will move outside in 0.3. But how does CO2 manages with low partial pressure? Oxygen has high difference of partial pressure to exchange. But how does CO2 manages? CO2 gets the support of the solubility. Solubility of the CO2 you say is 20 to 25 times higher than oxygen. And that higher solubility helps the CO2 to overcome the less partial pressure difference. So due to its high solubility, it can diffuse at a faster rate. So for CO2 exchange of gas, the solubility is helping. For oxygen, the high partial pressure gradient difference is helping. And for both the gases to rapidly diffuse from alveoli to the blood capillary and blood capillary to the alveoli, the diffusion membrane, the respiratory membrane is just made up of 3 layers, the alveolar epithelium, the basement membrane, and the endothelium blood capillary. That is hardly less than 1 millimeter, and this 1 millimeter less thickness is helping to have a faster exchange of gas. Are we clear? So exchange of gases done. Next, transport of gases. Oxygen transported 97% in the form of oxyhemoglobin. Solubility of oxygen is very poor. Sir, now NTA giving arrange the statement sequence, how to solve it. But obvious, Parida, it is a most commonly asked question. That breathing sequence question is very easy. Okay. And that was a statement based and multiple time it is asked. It is highly expected questions. Arranging the order. We have to remember that order. Okay. Okay. Next. 3% in dissolved state. Talking about carbon dioxide. Carbon dioxide 7% in the plasma. It is dissolved. Okay, solubility is higher, 7% dissolved in plasma. Remaining 20 to 25% is bounded to the hemoglobin in the form of carbaminohemoglobin. 70%, the major form of the carbon dioxide, is transported from the tissues to alveoli in the form of bicarbonates. But where are these bicarbonates formed? The bicarbonates are formed in the RBC, but they are transported in the blood plasma. And remember the enzyme also, carbonic anhydrase enzyme. Carbonic anhydrase enzyme. Carbonic anhydrase enzyme, okay? Carbonic anhydrase enzyme is present in less quantity in the plasma, but very high quantity in the RBC. Remember this? I hope you remember this. In the RBC, the quantity of carbonic anhydrase is very high, whereas in the plasma, it is very, very less. ठीक है? Are we clear, guys? ठीक है? That's all for this lecture, guys. I will stop here. Next class is there, guys, for you all. Any other classes there? After this, which class is there? अच्छा, ठीक है, ठीक है, ठीक है. Okay, guys. Okay, okay, okay. No, sir. No class na. अरे तो we can continue ना यार. So we can continue, no problem. Let's continue and finish the topic. Shall we continue? Tell me. Come on. Let's continue, yaar. Okay? Yeah. Let's continue. Let's continue. Vishnu Vardhan sir, where are the senior classes going? Senior classes are going offline also. Come on. So now coming to the oxygen dissociation curve. Okay, oxygen dissociation curve is telling you the relationship between the partial pressure of oxygen and the percentage saturation of oxygen with hemoglobin. So when you draw a graph, or when you plot a graph with the partial pressure of oxygen with the percentage saturation of oxygen with the hemoglobin, what shape of graph you are getting? You are getting a sigmoid curve. You are getting an S-shaped curve. But why you are getting an S-shaped curve here? Because the initial bonding is slow. The initial bonding of the oxygen with the hemoglobin molecule is slower. Okay, the first oxygen molecule takes a little time to bind, but as the first oxygen molecule binds, the remaining 3 oxygen molecules also binds very quickly. So initially we have a little slower rate, and then after that it accelerates, and then we get a asymptote stage or a plateau state when all the hemoglobin molecules are completely binded with the hemoglobin molecules. Are we clear with this? ठीक है? Will be completely binded with the hemoglobin molecules. So you are getting an S-shaped curve. Now, this curve can show 2 types of shift. This curve can show 2 types of shift. Tell me, what are the 2 types of shift? Rightward shift and leftward shift. One is the rightward shift and another one is the leftward. So one is the rightward shift, and another one we can say is the leftward shift. Another one is the leftward shift, and here is the rightward shift. Where is the rightward shift occurring, guys? The rightward shift occurs at the tissues, and the leftward, L for left, L for lungs. L for left, L for lungs, okay? So already the diagram which is shown is a leftward shift only. On lungs, the conditions are such that, what are the conditions in the lungs? Tell me, guys. In the lungs, what are the conditions? In lungs, there is high partial pressure of PO2. Yes, NCRT line. Binding of hemoglobin or binding of the oxygen with hemoglobin, the primary factor, it is primarily based on the partial pressure of O2. Partial pressure of O2 should be higher. Then there should be low partial pressure of CO2. There should be low partial pressure of CO2. H plus should be low. If H plus is low, pH is high. And what about the temperature? Temperature also should be low. Temperature also should be low. High oxygen. Remaining everything should be less. H plus and pH, they are inversely proportional. So H plus is lower, so pH will be higher. All these factors will lead to oxyhemoglobin formation. They are in the favor of association. They are in the favor of binding. So you can see at the lungs, all the favorable conditions are there that is helping the oxygen to bind with the hemoglobin. Whereas on the tissues, the conditions are exactly opposite. In the tissues, we will say there is a higher PCO2, low O2, high temperature, high H plus, and pH is low, and pH will be low. All this factor will lead to what? Tell me. All this factor is going to leave the oxygen. This factor will lead to dissociation, unbinding, dissociation. So we can see oxygen and hemoglobin have a reversible association or an irreversible association. Reversible association or irreversible association? Tell me, guys. Is it reversible or irreversible? It is reversible. What it is? It is reversible. At lungs, there is binding. At tissues, there is unbinding. At lungs, there is binding. At tissues, there is unbinding. Are we clear? This is how hemoglobin should transport oxygen. Yes or no? Yes. But there is some gas which makes a irreversible binding with hemoglobin. There is some gas which makes an irreversible binding. The condition is very dangerous. The person may die due to this. And that type of condition is called as what? What is that poisoning called as? What is that poisoning called as? Yes, the answer is CO, carbon monoxide poisoning. CO poisoning, CO poisoning, carbon monoxide poisoning. So when you are placed in a closed chamber or some faulty machine is there, some heater is there, faulty in winter season from the north, you might have heard, okay, some person died. Some heater, geyser had some problem that released the carbon monoxide. They were present in the closed chamber. So if carbon monoxide leakage is there, carbon monoxide is a colorless, odorless gas. So if it is a closed chamber, the carbon monoxide has a 200 times more affinity to the hemoglobin. So carbon monoxide is like a psycho lover, and that binds irreversibly with the hemoglobin and it doesn't leaves it. And what will happen? Your brain, your chemoreceptors only detect CO2 and H plus. Your chemoreceptors only detect CO2 and H plus. Oxygen role is quite insignificant. So CO they are not checking and CO is making the damage. Your tissues are going to die due to deficiency of oxygen. Are we clear? That is the carbon monoxide poisoning. So remember this. Can be asked as a question because many times it is asked in the PYQ. However, the binding of oxygen and CO2 is reversible. Okay, binding of CO2 and oxygen is reversible. Are we clear with this? Yes or no? Are we clear with this? Yes or no? Next, this line here. Every 100 ml of oxygenated blood can deliver 5 ml of oxygen under normal physiological conditions. But now my question to you. 100 ml of oxygenated blood normally carries how much oxygen? 100 ml of oxygenated blood normally carries how many oxygen? 100 ml of blood carries how much normally oxygen? So see, remember this. Every 100 ml of oxygenated blood, normally I am saying, normally it carries 20 ml oxygen. Normally it carries 20 ml oxygen. Okay, now under normal resting conditions, under normal resting condition, it will deliver how much? It will deliver means how much it will give to the tissues? It will give 5 ml to the tissues. Under normal resting condition, 5 ml. Now let's say I am in the gym, I am exercising, I am running, I am cycling, I am teaching you, I am standing for the last 3 hours. So now my muscles are exercising, I need more oxygen. So under exercising condition, how much? Under exercising condition, now it will deliver 15 ml. So just remember this. So initially, how much percent it gave? Initially, how much percent it gave? Initially, it gave 25%. Because 5 ml is 25% of 20 ml. But under exercising condition, how much it will give? Under exercising condition, it is giving 75% of the 20 ml oxygen from the 100 ml oxygenated blood. And you know what? This data I can show you on the ODC curve. On oxygen dissociation curve graph also you can see. So it means that under normal resting conditions, you got 25% of the oxygen. Where is the remaining 75%? Under normal resting condition, your tissues got 25% of the oxygen of the total, that is 5 ml. What is the remaining value? Remaining 15 ml went into the veins. So that is 75% in the veins. Are you getting me? Is this clicking with you guys? Is this clicking with you guys? Arey, is this clicking with you guys? So, what is the partial pressure of oxygen at the tissues? What is the partial pressure of oxygen at the tissues? At tissues, how much partial pressure of oxygen will be there? Yeah, it will be 40. In deoxygenated blood also it will be 40. In deoxygenated blood it is also 40. In deoxygenated blood also it is 40 only now. In deoxygenated blood also it is 40. Now look at the magic. Okay, I hope you get this magic. Look here. So, when I talk about 95 mm of Hg of oxygen, when I say 95 mm of Hg of oxygen, you will get 100% saturation. Roughly, I will say this is 95 somewhere here. At this level, when I check, there is 100% saturation. There is 100% saturation. All hemoglobins are combined. But what at 40? What at 40? Because at 40, out of this 100%, how much percent oxygen will be gone? Out of this 100% saturation, how much will be gone? 25% will be gone. So at 40, how much will be the saturation? 75. Is it coming like that? Is it coming like that? Yes, it is coming like that. 75%. Yes. So at 40 mm of Hg, either at your tissues or in the deoxygenated blood, 75% saturation is there. So what is the difference here? Oxygenated blood had 100% saturation, 95 mm of Hg. At tissues it became 40. So it has to leave the 25% to the tissues. So the remaining left will be 75%. Are we clear with this? ठीक है? So you should be knowing this indirect data, because on this indirect data, if they try to ask challenging questions, they might ask. In our exams also in the past, we have asked this type of questions. Are we clear? चलिए. ठीक है? Done. And same if they are asking for the carbon dioxide for 100 ml. Sir, at 30, there is 50% saturation? No. 50% saturation is at 22 to 25. 22 to 25. Okay? 22 to 25. At 22 to 25, you will see 50% saturation. That is called as the P50 value. Okay. Moving ahead, for CO2, every 100 ml of deoxygenated blood delivers 4 ml of CO2. Here, such calculation is not there. Here we are not plotting carbon dioxide hemoglobin dissociation curve. No, because the maximum amount of carbon dioxide is going in the carbonate form. So that's why you cannot plot a graph for carbon dioxide dissociation curve. For oxygen you can do, because 97% is carried by your hemoglobin. Are we clear? Regulation of respiration. Regulation of respiration is done by medulla, rhythm center, that is the boss. It is having the neuron groups which are sending the signals to your respiratory muscles, diaphragm and intercostal muscles. Then we have the pneumotaxic center. Pneumotaxic center in the pons, it helps to reduce the inspiration duration. Reduce the inspiration duration. So when you are reducing the inspiration duration, is your breathing rate increasing or decreasing? Tell me. Something you were doing in 2 hours, now I am telling you to do in 1 hour. Is your speed increasing or decreasing? Pneumotaxic center reduces the inspiration duration. So what happens to the breathing rate? What happens to the respiratory rate? Respiratory rate will increase. You will do shallower breathing. Fast breathing you are going to do. So pneumotaxic center reduces the inspiration duration, thereby altering the respiratory rate. It actually increases the breathing rate. Are we clear? Yes. Sir, what is chloride shift? Chloride shift is nothing but in the RBC, when the bicarbonates are formed in the RBC. Lot of bicarbonates move outside from the RBC due to higher concentration. Bicarbonates are moving outside, due to which the chlorides are coming in. As the chlorides are coming inside, what is called chloride shift? Chloride shift occurs at tissue level. The same chloride will go out. That is called as a reverse chloride shift. Reverse chloride shift that will occur at lungs. Are we clear? But asking about chloride shift, the chances are very less. Are we clear? And then we have the chemosensitive area. Chemosensitive area detect H plus and CO2. One present in the medulla, central chemoreceptor, other in the carotid and the aortic bodies. Are we clear? And the role of detection of oxygen. Role of oxygen is quite insignificant. That's all. Diseases. You go through the diseases. Just remember, emphysema is a chronic disorder. Chronic means which develops over a long time period. Acute is a problem which develops something very shortly. Okay? In quick time, some problem develops, that is acute. Emphysema is a chronic disorder. For occupational respiratory disorders, remember silicosis, asbestosis, this word. And here, in this, there is a question, this one. I hope you know what is hypoxia. Have you heard of hypoxia? What is the meaning of hypoxia? This question is important here because hypoxia is not explained in the NCERT lines. Hypoxia is low level of oxygen in blood. So if you go to high altitude, oxygen is less. So that is called as hypoxic hypoxia. The oxygen already is less in the air, so you are getting also less in the body. So that is hypoxic hypoxia. If you are anemic, you have sickle cell anemia, thalassemia, iron deficiency anemia, then your oxygen carrying capacity will be decreased. It will be called as anemic hypoxia. And one question, when person died due to cyanide poisoning, the cyanide damages your respiratory mechanism, the Krebs cycle and all is completely damaged. So the cell dies due to toxicity of the cyanide. So it is called as histotoxic hypoxia. Histotoxic hypoxia. I remember some question was asked, what are the different types of hypoxia? Many students were saying, sir, why this question? But this question is related to NCERT. What are the types of hypoxia? You can go and check and just write down that points. Hypoxic hypoxia, high altitude, low oxygen. Anemic hypoxia due to anemia conditions, low oxygen. And histotoxic hypoxia due to poisoning. Okay, that hypoxia is occurring due to cyanide. Fibrosis is like when a person is working in silica industry, asbestos industry, the particles are very fine, they get deposited in the alveoli. Your immune system tries to fight, but it doesn't destroy the non-living molecule. What happens over the period of time? So much damage has occurred in the alveoli, normal tissue is gone. Now fibrosis has occurred, collagen fibers are deposited like wound healing, scarring. Like you get the scar, so that is pulmonary fibrosis. That alveoli is not useful for exchange of gases. Okay, Cyanosis is something else. Cyanosis is bluishness of the lips and the fingers. When due to low oxygen, your lips and fingers turns bluish in appearance, that is cyanosis. Okay? Cyanosis is telling you that person is having low oxygen in the body. Okay? Chalo guys, so that's all for this class. Thank you for waiting. Okay? So bye-bye. See you in the next class. Sir, do we need to revise the notes? I think for biology, NCERT is enough. NCERT, all the lines, summary, questions, just go through the NCERT. That is more than sufficient. And whatever exam questions we are asking, I think it is more than sufficient. And Gosalites is covering all the different types of questions. So many tests you have given, it has covered all the questions. Now also we are going to cover all the questions. So just read NCERT. That is more than sufficient. Okay? Bye guys.